all over me. Well, I've been there before. All right, guys, I'm trying to get you guys on camera. I'm trying to get everybody wearing their wonderful scrubs. Got the scrub jacket orders from almost all of you. If you haven't ordered a scrub jacket by this point, you better tell me soon, but that was on you. And I refuse to go hunt you down for it because, hey, I've got to hold you guys a little accountable. This is the whole, hard knock life type of approach to things. All right, guys. The goal with today is to see how comfortable you are with the simplified model of metabolism, which means we've got our good old Nike swoosh here, and it's your turn to help me fill it out. So I'm going to start, and I'm going to literally progress across the metabolism of an individual moving in a left to right fashion. But start me off, everybody. What is the definition of metabolism? How do you define metabolism? What do you got for me? I define it in a specific way. I just don't know how you guys define it. I mean, I'm guessing you guys could use metabolism in the sentence. So when you talk to somebody, it's like everybody knows what it means, but nobody always knows. So let's see, chemical and physical workings of a cell. All the chemical and physical workings of the cell, reactions, like these answer, chemical reactions. I'm going to go with chemical reactions in a living system. Okay, so it's all of the chemical reactions happening in the living system. Some chemical reactions start by breaking things down, and so half the story is true, Lisa. Breaking down of things is the catabolic side, and then the anabolic side is the building up of things. So if we were to take this little chart right here and separate it into the two workings, when the swoosh goes down, the molecule sizes are going down. So this will be our catabolism on this side. And then as they hit their lowest point, they start to build back up, leaving this other side over here for our anabolic reactions. And so our metabolism is a combination of both. All right, guys. So we've got catabolic and anabolic reactions happening, and the swoosh lets us consider the size of the molecules. So we start the swoosh at a midway size, right around a building block size. And for all the metabolism in a living system, we all start with this preferred molecule. So what's the starting molecule of your metabolism? There's a hint. It tastes sweet, everybody. All right, so it's going to be glucose. So one feature right off the bat about glucose, everybody, is this is a carbohydrate. And I know that carbohydrates have a 1-to-1 ratio, which means for every one carbon, I have 2 hydrogens and 1 oxygen. Well, it turns out I have 6 carbons, so it's a 6-carbon molecule, which means how many hydrogens do I have? If it's a 1-to-1, that means I have 12. Oops, not 23. And then the last one would make me 6 oxygen. So really, guys, I'm looking at a C6 molecule. That's kind of decent in size, a little bit of a larger molecule. Now I'm going to go through a series of reactions, and here is my question. Only 4 of you even attempted CK12. So what did the rest of you do to get ready for metabolism today? What did you do to prepare yourself to know about glycolysis, Krebs cycle, and the electron transport chain? So, ooh, the picmonics are good. They are good. What else we got? Anything else? Glycolysis. Nice. Picmonics, Krebs cycle. But you guys will also see it as the TCA, citric acid cycle. Electron transport system or electron transport chain really depends on the one, so the big 3. And then, of course, if oxygen is not present, you'll also see fermentation here. Okay, guys, so these processes, some of you use picmonics. Now, that was literally 2 of you that answered my question. So how about the rest of you? Yeah, I know who did CK12. How about the rest of you? You guys were tasked with homework, non-homework. There's a whole lot of the rest of you that have some splaining to do right now. Scientist Cindy, she's rocking it, man. I love her for this section. I should put all of you on camera and question you right now, like depose you, see who's telling the truth. The answer is, I didn't do anything, Dr. C. The answer is, I totally forgot I was supposed to do this. All right, for the rest of you, honestly, got the CK12, buffered the video. Okay. Yeah, I saw that, Lisa. I did actually see that you got one done, though. She got one. One is better than zero, which is what's happening for the rest of you. And I know which 4 of you went into CK12. I'm very proud of you for listening to my helpful request. I promise it's going to help you. But for the rest of you, I'm not bad. I'm just disappointed. Yeah, it's a good one. It's a good mom thing. Now, you guys are definitely going to want to capitalize some resources as you study for this next exam. Today, we'll go through, break these down, but it is going to be one of the videos you'll probably watch twice, guys. It is definitely a more challenging concept. I do consider Chapter 8 the hardest chapter in your textbook for you guys. I think Chapter 9 is the second hardest, so next week is going to be equally as fun, okay? So I hope you guys get a chance to do this, but you were supposed to have done it before we came in today, so blah. All right, guys, now we start with the sugar that feeds us all. And you know what? You as a human use glucose as your primary source of energy. Primary. Now, it's possible that we can incorporate other types of metabolism like lipids, and worst case scenario, you can actually break down proteins for energy if you need them. That happens when individuals are starving, and we'll be learning about that over in the A&P world. But glucose is broken down through these series of reaction to some important precursor molecules that we can turn around and rebuild. So help me out, guys. What are those precursor molecules? Okay, so one of them is acetyl-CoA, and that's actually a 2-carbon sugar. It's acetyl group, which is a C2, guys, and it has 2 carbons. So it's obviously much smaller than glucose, which was 6 carbons. All right, then there's pyruvate, my good old buddy pyruvate. That's actually a 3-carbon sugar, so it's much smaller. And then we also have glyceraldehyde 3-phosphate. Okay, and that one also is a 2-carbon sugar. No, it's a 3-carbon sugar. All right, but we're not going to worry about G3P too much, guys, because we're going to focus on these other 2 as a key way. Now, when we get past that, guys, precursors start to be used to build other things. And right about the same size as glucose over here, we build up those monomers, sometimes called those building blocks of our metabolism. So we've got the monomers. This should be throwback to Chapter 2. You don't even need me for this, guys. So help me out. What are those building blocks? So the building blocks will be not carbs. They're going to be, okay, so I'm going to go with amino acids. I'm going to go with what type of carbs, everybody? I'm going to go with the carbs being, okay, so monosaccharides, sugars. So monosaccharides are the sugars that we talk about. When it comes to DNA, let's do my DNA next really quick, guys. What would those be? Those would be what we call nucleotides, and those are going to include those A, T's, G's, and C's, and don't forget U in case we're building some RNA, leaving that last one, a group of fatty acid chains. And these guys, those fatty acids, you know, those are either saturated or unsaturated. Okay, so these building blocks are literally what we're going to use to start making the major molecules of a cell. And the major molecules of a cell are polymers of these building blocks. And so we think of these polymers as our macromolecules. And in fact, you find these guys on a food label because these are the molecules that you consume, a little bit larger, that give us a lot of energy. All right, guys, so the macromolecules that come out from amino acids are what? It's going to be our proteins. And an example of a type of protein for you guys might be some enzymes, okay? So we learned about function of enzymes, but obviously those are crucial for our microbes. You guys may remember proteins made up 50% of the dry weight of an E. coli. That's a lot. Next one. Monosaccharides combine with other monosaccharides to form
polysaccharides.
Polysaccharides, thank you. And some example of polysaccharides in your cells, guys, we might see some surface markers like glycoproteins, but when we're talking about it in microbes, we're looking at cell wall structure. So like the chitin that we talked about before, we're talking about the peptidoglycans. That word glycan is letting me know I've got some sugars. Even lipopolysaccharides would be an example, LPS would be an example that we find in bacteria. All right, guys, now monosaccharides form polysaccharides, but what about nucleotides? Nucleotides combine to make the two main genetic materials.
Nucleic acids.
Sure, which are DNA and RNA. So those are our key ones, but we can't forget our good old buddy, also ATP, because we're going to need some of that energy in a little bit. Now, lastly, guys, we're going to have to worry about fatty acids forming what structures in a cell?
Cell membrane.
Okay, so they're going to be complex fats like those in cell membranes. So I'm going to just go ahead and go phospholipids for you guys. I am tired today. Waxes like those we find on the surface of mycolic acid, like mycobacters. What else, guys? About lipopolysaccharides again. So these guys are going to form these larger structures, and it's these larger structures that combine, and even though bacteria don't have organelles, these larger structures help to form a total cell. And so the cell is at the top. And if we're talking about a cell, guys, the types of cells we talk about here in the world of microbiology are going to be prokaryotes. And we do spend a little bit of time learning about our eukaryotes along the way. So prokes and the eukes get us started. All right, this simplified model of metabolism is easier than you think if you just walk it through the breakdown and the builds up. But metabolism is never just one. It is never just breaking things down. It is also building things up. You literally eat calories so you can do this all day, every day in your cells. Sometimes enzymes are working 10,000 reactions per second, and you have millions of enzymes in each of your cells. So you guys, this is a magical set of chemistry experiments happening inside you at every given moment. All right, guys, so what we're going to do right now, we're going to take this metabolism and we're going to put it into action. But I hope this summary slide experience gave you an opportunity to just put your brain in there. And guess what? In the middle of a quiz, the Swish could be a useful tool to help you study. The Swish is going to help you guys that are in my A&P class as well, because this Swish is really going to build the foundation for what we learn about in that body set of body systems as well. Okay, guys, so there was a method to the proverbial madness. I like what you guys did here. I'm going to delete the old one. All right, anabolic. I'm just going to add one thing to it so it's absolutely perfect for you guys. The reactants are what to the products? I'm talking about an anabolic reaction. The reactants are
Less than.
Okay, less than the products. Which means in a catabolic reaction, the reactants are greater. All right, guys. So, oops, I switched those around. Guys, see, I don't know what's going on with me. Ooh, old gal is just tired today. I do know how to do math and do less than greater than. All right, so we are going to sneak ahead and start by actually incorporating those metabolic pathways. And you guys remember that we spent the last part of our class talking about the function of enzymes. So as a brief review for you guys, we talked about the different types of reactions, and we only had one type of enzyme that we've already begun our journey to investigate, which were some of those oxidoreductases. These guys, like the Leo the lion, help to perform oxidation reduction reactions. Lose electron is oxidation, things get more positive. Gaining an electron is reduction, and things become more negative. There are a set of enzymes that are used in microbes where they spit them out. Those are exoenzymes, where they keep them internally. Those are endoenzymes. Now remember, microbes, especially bacteria, guys, they are so tiny, and energy conservation is so important that they don't keep things that they don't need. So constitutive enzymes are enzymes that they know they're going to need all the time, but regulated enzymes are enzymes they turn on, induce, or turn off, repress, depending upon the available amount of substrate. And that really affects how they monitor and maintain their energy needs. Now, we know that we can do a lot with our knowledge of enzymes. Enzymes have those active sites. They're folded just appropriately. So we had the apoenzyme and the different cofactor coenzyme creating a holoenzyme. However, if we want to mess with some enzymes, a couple of things we can do. We can do competitive inhibition, where we literally battle for an enzyme's activity at the active site. We can also do non-competitive inhibition, battle for an enzyme's activity by binding to a site different than the active site, and it changes the shape, or what we call conformation of that enzyme. At the end of the day, guys, we're designing medication oftentimes to affect the activities of the enzymes inside a bacteria. Or when you go to cook food today, you're actually performing that cooking process when you apply heat with the intention to denature the proteins, which you're seeking to denature the enzymes. If you're worried about cooking your meat to a specified temperature because you want to break down the enzymes in that bacteria. I mean, the next time you guys go to cook your meat, you're like, oh, I'm breaking down enzymes. Like, that's really what's going on here. I know you're going to have a new world order when it comes to looking at why you cook food. All right, guys. Now, at the end of the day, enzymes are playing an important role in driving the processes happening inside your cells. And there's a lot of processes happening. So right off the bat, guys, some of the key things that are happening inside your cell are what we're going to break down. But to get there, we got a little bit of pre-learning to do. And I'm going to have you guys hold some purses and things like that, okay? I'm also going to introduce to you the major concepts of... Oops, sorry guys, I just don't like having a little writing on there in advance. Okay. I'm also going to introduce you the major concepts of energy buildup as well. But let's go ahead and make some energy in a cell. We're going to be talking about bacteria today. And then when you guys take my A&P class, if you are in that class with me, that's when we'll talk about you. Okay. So for a little bit, guys, don't be so selfish. Let's talk about bacteria. Okay. All right, in bacteria, remember, they have a very tiny cell they got to maintain, but it's them against the world. And their energy needs have to be met at every moment or else they die. Okay, so there's a series of combined reactions. These are the same things we could say about catabolism and anabolism. And the reason why we couple metabolism together is we need to maintain the right amount of energy to survive. You guys may remember right when I first showed you it that we lose about 60% of our energy to heat. What a waste. Oh my gosh. But we need to release that energy somewhere, so heat takes a good chunk of it. Along the way, though, exergonic reactions, which are reactions that give off energy, sometimes called exothermic reactions, since the energy they release is oftentimes released in the form of heat, these guys are going to make the ATPs. Then it's those reactions like those anabolic reactions that are going to need the ATPs. And so endergonic, or sometimes endothermic reactions, we call them, which are the same thing as anabolic reactions, require that energy. Okay, guys, so you have some reactions that give it off, other reactions that use it. Some reactions that give it off, other reactions that use it. Some reactions that give it off, other reactions that use it. And you balance between that all the time. And your bacteria is doing the same exact thing, trying to balance between the reaction types. So we couple these reactions to make sure we can fuel a cell. We start with something like glucose with a whole bunch of high energy bonds inside it. And as we break it down, the end result of all of our metabolism is to produce some CO2. We've lost all the bonds. We've lost 6 major bonds over here, giving off 6 CO2s. And along the way, as we broke that energy apart, we got a bunch of ATPs. This is the purpose of all of your metabolism, is to pop out a bunch of ATPs. Now you got those ATPs, so you can turn around and grab those precursor molecules and build them back up. Now, the major type of reaction we're going to talk to you guys about throughout this are a set of reactions that break things down and build things, and then we'll go at the very end and just talk about building some things up. But we do want to make sure that you guys just recall how Leo the Lion says grr, because these important redox reactions, reduction, oxidation, coupled reactions, are going to be a major part of the metabolism we learned about today. Okay, guys, so don't forget, we have a couple of taxi cabs. I already mentioned our friendly taxi cabs. One of them is NADH, and another one is called FADH2. These taxi cabs are going to carry electrons for us, okay? And we're going to show how they perform a type of ATP production that is specialized to those electrons. Now, just a friendly reminder, here's your ATP. ATP is a high energy molecule. It's a high energy molecule because it has some very high energy bonds. One bond right here and one bond right here between the adenosine tri or the adenosine diphosphate. These two extremely high energy bonds can be made and then broken, and then made and then broken, and then made and then broken all the time, forever and ever and ever. And that's how you survive. I know it sounds absolutely crazy to think that your energy is completely derived in chemical bonds, but that's how we do it, everybody. You turn the lights on because you make and break these high energy bonds. So basically, guys, what's going to happen to ATP is we need to have phosphate so that we can pop it off or put it back on. One type of phosphate availability, it comes from different substrates. Maybe you have a protein out there that holds onto a phosphate. That phosphate can be removed off of something else, donated to ADP, and you make ATP. So this type of donation is known as substrate level phosphorylation. All right, guys, now this is a hard concept for students to learn because you're trying to imagine a molecule that is just holding on to phosphate for you. Okay, guys, so I'm going to take you on a journey. Let's go shopping. Let's go shopping, okay? So we're going to go shopping, and I'm going to pretend that I'm like Julia Roberts in Pretty Woman level shopping, like I have money. I don't have that money, but and no longer I just online shop now. That's like my life's existence at the moment. But all right, guys, we're going back circa the 1980s. We're Julia Roberts minus the extracurricular activities in the nighttime, and we're going shopping on Rodeo Drive. Okay, guys. So the whole thing is whenever you go shopping, you're looking at stuff, you're shopping around, and you might have to have somebody hold your purse for you, right? So you ask somebody to hold your purse, then you start buying some stuff. So you exchange that purse with your purchases, right? Okay, can you hold my purchases for you? And you guys may remember that when Julia Roberts walked into the hotel after her day of shopping, she had a whole bunch of people following her with all of her purchases. Oh my God, they were holding on to her purchases. It was amazing. Well, here's the whole thing, guys. If you ever take somebody shopping with you, sometimes you have to have some, hey, can you hold my purse for a second? So that they can hold on to it, and then they give it back to you so you can pay for your purchases. And so we are on this shopping trip of our lives, and what we're doing is we're asking molecules to hold our purse for us. We're asking molecules, hey, can you just hold on to this for a second? I'm going to need it back in a moment. Okay, and that's all they're doing in substrate level phosphorylation is holding onto our purse. So the ultimate shopping trip, guys, they're just holding onto our purse or holding onto our purchases, and when we need it, we ask for it back. And that's how ATP is given this phosphate and loses the phosphate. Then you know what? Eventually, guys, this reaction will go the other direction. ATP will give off a phosphate and a substrate will grab it. Then we'll be like, hey, can you give it back to me? And then ATP. So you literally go back and forth and back and forth for your entire existence. And thankfully, we have one type of phosphorylation called substrate level phosphorylation to hold our purse for us. All right, guys, now let's say that shopping trip was so extensive that you needed to just send your packages back home. Okay, guys, so in this particular case, you know what you might do? You might load up all your packages into a taxi cab and have the taxi cab take them for you. Okay, guys, the taxi cab in this particular scenario is going to be those NADH and FADH2 molecules, because this is a type of phosphorylation called oxidative phosphorylation. So substrate level phosphorylation, hold in the purse. Oxidative phosphorylation is a type of phosphorylation where a carrier holds onto your phosphate for you, like NADH or FADH2. So oxidative phosphorylation is going to use those taxi cabs we talked about. Now, you guys, I'm sad to say only some microbes and things like plants can perform this third type. I'm going to tell you right now, I'm not testing you on this third type, but I am going to show you why this third type happens, because without it, you would cease to be able to live on this planet. It is that big of a deal. This third type is called photophosphorylation, and in this particular case, the sunlight is giving you the phosphate. I know, it's amazing, but the sun is like, here, let me give you the phosphate. In this particular case, it's only going to happen in organisms that can do photosynthesis. So photophosphorylation, the third one, kind of a cool one to sit back and learn about, guys, not necessarily one you need to write all the notes on, but photophosphorylation lets the sun hold your purse. In this particular scenario, the light from the sun gets trapped in a chlorophyll molecule found inside a chloroplast inside eukaryotes, or just floating around the inner cytoplasm of prokaryotes. When the sun's energy gets trapped in this really green pigmented series of molecules, that actually excites the electrons. Then the electrons get shuttled down an electron transport chain. As you move electrons around, you can generate ATP. And this is the simplest way I could possibly explain to you guys photosynthesis. The sun traps the energy gets harvested in the form of electrons, and electrons get taxi cab throughout the electron transport chain. All right, that's really cool. And this is literally how we produce light. Some of the neat byproducts of all this cool stuff are we produce oxygen, which we use for our respiratory intake. So there's some really cool stuff to this, but guys, I'm not going to hold you accountable to this. All right, now there's a series of reactions though that are happening every second on our planet. Did you know we have light and dark reactions? When the sun is out, plants help to produce oxygen to survive. When the sun goes down, the plants can still continue to produce oxygen even without the sunlight. Oh my God, it's amazing the evolution that's happened. So we have a bunch of different photosynthesizers that provide us with our oxygen. And guys, about half the O2 in the environment around you is due to microbes. There are bacteria around our planet. You guys, these are fields of bacteria and they have different colors. You see how these different colors are in place on these fields? These different colors are different chlorophyll molecules. There's yellow chlorophylls, there's orange chlorophylls, and there's red chlorophylls, as well as green chlorophylls. So if you've ever heard about red algae versus yellow algae versus purple algae versus green algae, these fields themselves are really just photosynthetic fields. It's amazing, but thanks to these, you're breathing today. Go for it, Waverly.
So CP12 said that photoautotrophs are what create 99% of our energy supply. So does that mean oxygen, and what does that exactly mean?
Okay, well, let's go ahead and break down photosynthesis for you. When it comes to photosynthesis, the way that these are producing energy, I'm going to go over here and I'm going to get my cool little, and I'm going to change it from red because I'm really burned out on red, guys. Ooh, we'll go magenta. Okay, guys, so here's what's going to happen. You got chemo autotrophs like you and me. And what do we need to eat? We need some glucose. So here's glucose right here on one side of the reaction. So whenever I'm hungry, guys, I remember glucose is my source of energy. And what do I need to survive? Well, since I happen to also be an obligate aerobe, I need some oxygen to survive. My body is made up of mostly water, and thankfully I got a lot of water to perform some of these reactions. Well, these are the needs that I have, okay? But along the way, guys, it turns out that if I want to take that glucose and use it and eat it and consume it, I got to get it from somewhere. And you know where I get it? I get it from photosynthesis activity in plants. What a plant's going to do, and you've heard this rumor that you can talk to your plants and it makes them happy and grow better. Well, when you talk to your plants, you're shooting some CO2 directly at them because you're breathing out CO2. And along the way, they will take that CO2. They will also take some extra water molecules. They take about 6 water molecules, and they harvest the energy from the sun. And the byproduct of photosynthesis is actually carbohydrates and oxygen. And so the byproduct of photosynthesis is the sugars that you and I need to survive and the oxygen that you and I need to breathe. So photosynthesis literally produces what we need to survive. And so how do we manage this? How do we deal with this? Well, guess what, guys? We eat plants. So if you eat your vegetables and your fruits and your veggies, you're consuming carbohydrates. That's the moral of the story. Everybody who's ever done a macro diet knows where do you get your carbs? You get them from potatoes, you get them from grains, you get them from plants. All right, but if you are a metasaurus, not a carnivore, but you're a metasaurus, you're one of those people that's like, I don't like veggies. How am I alive? Well, guys, guess what? Your animal that you eat, if you're a metasaurus and it's a cow or whatever it may be, guys, the animal that you ate these carbs. So the cycle of life and the recycling of nutrients came from the fact that the animal that you ate these carbs, gave it the energy, and then you ate its flesh for energy. Mind blown. Do you guys know why I have a fire behind me? I have a fire behind me because that's your body's metabolism. That is a fire. You guys are burning up inside. Remember, metabolism is a series of these reactions. So when that text said it, yeah, the answer is plants can self-sustain themselves. You know what plants do? It is so cool. Inside their eukaryotic cells, they have little tiny mitochondria to harvest energy. And then on the other side of the cell, they have chloroplasts to collect energy from the sun. So plants are self-sustaining autotrophs because they can grab the sun's energy and then they can make their own carbs. Then they can eat their own carbs with them and produce the necessary ATP to survive. Plants can literally just survive off of your CO2 and a little bit of sunshine, plus some water. Did you see the water in the metabolism? So plants can literally make their own energy and then eat themselves, I guess would be the easy way to consider their survival. Cool stuff. I don't want to be a plant. Plants are kind of lame. I mean, short of like a Venus flytrap, I don't want to be a plant. But other than that, hey, and you know what's really cool about Venus flytraps? Venus flytraps can digest like an animal. Oh, it's so darn cool. They shoot enzymes and dissolve the fly and then turn around and let their mitochondria digest, and it gives them more energy and they grow bigger. So by the way, if you've ever gotten yourself a Venus flytrap, I had a hard time keeping my Venus flytrap alive. I killed it. I'm not even gonna lie, guys. I'm a plant lover. I'm a planter. I've got a decent green thumb, and I killed my Venus flytrap because I did not move it out of the windowsill before it got cold. It was like years ago. go. So you know how you keep them happy though? Feed them a fly every once in a while. Yeah, just give them a nice little fly treat. It is the neatest thing you've ever seen because they go like this. They go, and they sit open, and then all of a sudden, and I mean, you can't watch them for that long. So then all of a sudden you come back and you're like, where'd the fly go? And it's inside the Venus flytrap. It's amazing. Oh my God, it's the coolest thing ever. I don't know what's wrong with me, but I really do think that's the one plant that I'm kind of finding okay. All right, nonetheless, the answer to your question hopefully was given. And so phosphorylation events are really an important thing to know about. But let's do a summary to where we're at, everybody. Exergonic versus endergonic reactions, we know the differences. The money molecule of your cells is ATP. We did a review of what it's made of, but we know that ATP lives in this recycle between ADP and phosphate and then ATP, and you literally go back and forth between this cycle. Inside your cells, the two main ways that you get those phosphates, one, you ask somebody to hold your bag. So substrate level phosphorylation, you use a substrate to hang on to your phosphate. Oxidative phosphorylation, you ask somebody to deliver your bags with taxi cabs. So those will be those taxi cab delivery services of the NADH and the FADH2, because those guys are going to taxi cab the electrons for us. And we'll learn how this happens. So as much as I introduce the concept, we're going to see it in action next. Photosynthesis, I just wanted you guys, we reviewed it, so cool, but you totally don't need to know this. You're welcome. I have taught microbiology for other institutions that still require nursing students to know photosynthesis in detail. I gotta tell you guys, I love photosynthesis. I had to take an entire class. I was a plant biologist a million years ago. When my undergrad, which does feel like a million years ago, guys, my undergraduate research project was on Lotus japonicus. It was on plants and plant nodules and symbiotic relationship with bacteria. And we published an article. It wasn't me. I wasn't primary author because I was an undergrad, but I gotta say it was one of the coolest things that got me to want to be a scientist, and it was all about plants. So I do like them, but I don't want to teach them. All right, guys. So nonetheless, let's go in and watch this ATP happen. And we're going to get there through a series of catabolic pathways. We're going to make our ATP via catabolism. Now, here's the coolest thing about glycolysis, everybody. Glycolysis is the starting pathway for everything. Glycolysis starts everything. It starts metabolism in the presence of oxygen. It starts metabolism in the absence of oxygen. These two over here are the no O2s, and this is the oxygen is present. So you know what we actually say about glycolysis? We say glycolysis is a ubiquitous pathway. And we know what ubiquitous is. It's everywhere. Well, glycolysis is one of the if it ain't broke, why fix it thoughts I have. All right, guys, so listen, you can believe your own formation of the universe. I would never want to step on anybody's toes, but as it stands, glycolysis is present in almost every single organism on our planet. This is the primary mechanism that they use for their metabolism. Like when I say almost every, it's like 99.999999999%. I have yet to give you a single organism that doesn't use a form of glycolysis. It doesn't always use the whole set, but I have yet to find an organism that can't do some form of glycolysis. So since that's pretty decent evidence that all organisms came from our good old buddy Luca, a last universal common ancestor, we just have to look at the difference between those organisms that lived in a world with oxygen and went one direction to make their ATP versus those organisms that live in a world without oxygen. And you guys remember this because last chapter we learned all about obligate aerobes through the spectrum to obligate anaerobes. Well, guys, obligate anaerobes still need metabolism too, but how they make it happen is much different than how aerobes are. And what are you again? Remind me, guys, what are you? You're an obligate. You're chemo, heterotroph, and an obligate. You're a mesophile. You're an obligate aerobe. Without oxygen, your existence is like, and you got less than a minute in you guys, right? So as it turns out, you cannot make it happen without oxygen. So you are going to need this pathway. At the outcome of this pathway, guys, though, check it out. 36 to 38 ATPs. In fact, guys, eukaryotes like you and me, we can actually get 40 plus. Oh my God, dude, we have some really cool, efficient systems, but we're getting about 36 ATPs. I'm going to tell you guys right now, we're not going to worry too much about this one in the middle, but the ones that come with anaerobic are much lower. Okay, so breathing oxygen gives you about 36 ATPs. Not breathing oxygen gives you about 2. Which one do you want? I want to breathe oxygen. I mean, you're like, oh my God, well, that's like clearly efficient. Which one are you investing in? I'm investing in aerobic metabolism. All right, guys, so those of you that take my A&P class, you're thinking about the respiratory system right now and the fact that we know our cells release CO2. Where does that CO2 come from? Well, that CO2 comes from your body's metabolism. And so if you are a human being and your CO2 is controlled by your respiratory system, it's because of this series of pathways. All right, I'm about to do it. Right in front of you on your study guide, you have a blank area for glycolysis. I've also got a mapping system for all of this. So just hold on tight, take a deep breath, and here we go. All right. Okay. So if you did not do the pre-planned pre-homework assignment thing I told you, you're about to have a heart attack right now. If you did, you're like, okay, I saw this in there. I kind of remember this one. Well, I want you guys not to memorize a single step in this pathway, but follow along with me, guys. What did we start off with? We started off with a 6-carbon glucose molecule, C6H12O6. And the steps of glycolysis utilize a series of enzymes to make this happen. Okay, so we've got a series of enzymes that make each of these steps possible. And I can actually, let me sneak ahead really quick. I can show you the enzymes. I think I have them listed on one of these. No, I guess I don't. You're in luck. I don't have any of the enzymes listed, but each of these arrows uses an enzyme to help it function. But as a product of that enzymatic action, these are what happened. First step, guys, remember glucose is C6H12O6. Well, guess what I do? I ask glucose to hold my purse. Check it out, guys. Glucose is going to hold my purse. There's a phosphate. It's holding my purse. And I'm like, hey, glucose, hold my purse for me. Then I isomerize glucose to fructose. And then I'm like, hey, fructose, do me a favor, hold another purse for me. So now you've got this molecule that is holding on to 2 phosphates for you. Substrate level phosphorylation. You see that happening right here, guys? All right, so then I go ahead and I just split this molecule into 2. And now everything that's going to happen is going to happen in duplicate. So both of them are holding onto a phosphate, and then I go through a series of reactions. The first thing I'm actually going to do, I'm going to produce a few taxicabs. So I've got 2 taxicabs right here, 2 NADHs. And I'm going to start making some stuff. Okay, guys, so you ready? Ready? Look at this with me, guys. I used 1, 2 ATPs. I use those ATPs. But right here, I'm going to produce 1, 2 ATPs. So I used 2 ATPs, minus 2 up here. But I made 2 right here. Then I keep going. I do some cool isomerizations. I release some water molecules. And next thing you know, guys, I'm actually going to make 2 more ATPs. So at the end of this, what I end up producing is a molecule that has 6 carbons in it, and we call that pyruvate. And I made 2 pyruvate molecules. Okay, so this whole pathway led to this. Let's go summarize it together. The steps of this pathway are broken down for you guys. I can show it to you again if you need to see the steps, but what you're most excited to see, guys, and here's what you need to be writing down, is the net reactions, because the steps are not what I need you to know. So let's start this reaction, guys. Glucose plus 2 ADPs, because we needed to have the foundation, and 2 NAD plus molecules ended up producing 2 pyruvates, 2 net ATPs and 2 NADHs. Okay, so the net reaction itself was glucose made 2 pyruvates, 2 ATPs, and 2 NADHs. That's the net reaction. All right, glycolysis leaves behind 2 pyruvates. Those pyruvates can actually be put into other pathways, but we start with an intermediate step where we take those pyruvates and convert them to something known as acetyl-CoA. Now, if I want to take pyruvate right here, guys, I can go down one pathway and actually enter fermentation. I can also take that pyruvate and build it up if I need. So here's the neatest thing, guys. Pyruvate is a precursor to a lot of different things. If I'm going to continue with metabolism, pyruvate goes this way. If I'm going to continue to build my cell, pyruvate goes that way. If there's no oxygen in the environment, pyruvate goes this way into fermentation. So pyruvate is a central molecule where lots of things can happen. And what we got to do if we're going to continue metabolism, guys, we're going to take that 3 carbon, 1, 2, 3 carbon sugar, and we're going to make an NADH, and along the way we're going to attach a coenzyme A and make this into something called acetyl-CoA. And acetyl-CoA can also act as a central metabolite if necessary. So what happened is glycolysis and that cool little intermediate step have led us to this point. You ready, guys? Here's the summary. First step, glucose started us off. We formed 2 pyruvates, which were C3s. We also, at the end of glycolysis, formed 2 ATPs and 2 NADHs. Then we had that intermediate step. How many pyruvates did I take into the intermediate step, everybody? I took 2. So twice I made 2 acetyl-CoAs, and those ended up giving off a carbon. I went from C3 to C2. I lost a carbon. I lost a carbon. I lost a carbon. My carbon structure is falling apart. Well, guess what? You released that carbon in the form of 2 CO2s. So for the first time ever, guys, you've generated CO2s. Your cells are breathing, everybody. And along the way, this intermediate step gave us 2 more NADHs. So this one is glycolysis, the big G. This one right here is just a step in the middle to get your metabolism ready. Those of you that did your pre-assignment, what's the next step though? If oxygen is in the environment, an aerobic molecule will go to the If oxygen is in the environment, the presence of oxygen goes into the Krebs cycle, everybody. All right, guys, so what do I have going into the Krebs cycle? I've got acetyl-CoAs going into the Krebs cycle. How many acetyl-CoAs do I have? You guys, it was just on the last slide. How many acetyl-CoAs do I have? I got 2. So I'm going to go through this Krebs cycle twice. You ready? Is everybody ready? Hold on to your socks, everybody. Here it comes. Whew, here's the Krebs cycle. All right. So there's the acetyl-CoAs. They came from the pyruvic acid, and along the way you had those 2 acetyl-CoAs. Don't forget, guys, you made your first CO2 up here, so your cells breathe it. The acetyl-CoA is going to come in and enter the Krebs cycle, bind itself to the oxaloacetate, and become citric acid. And suddenly, this 4-carbon oxaloacetate now has 2 more carbons on it because you added the acetyl-CoA, so it's now a 6-carbon sugar. Then it becomes isomerized to isocitric acid. And then in this next step, you actually are going to lose a CO2. So now you went from being a 6-carbon to a 5-carbon. You're a C6 over here, now you're 5. And one of the carbons you brought in is lost. Along the way, you made a taxicab. Then you got a 5-carbon, and guess what you'll do again? You'll lose another CO2. Now you got a 4-carbon, and you make another taxicab. Then you keep going a little bit more. You go from succinyl-CoA, you drop off that coenzyme A, and you make an ATP. Now you're still a 4-carbon sugar, but guess what, guys? You can do some cool isomerizing and you can make a different version of a taxicab right here. Along the way, you keep going, you're in malic acid, and you're going to go ahead and make another set of taxicabs before you end up at oxaloacetate. And then guess what? Since you had 2 acetyl-CoAs, you go through everything again. And along the way, you make another CO2, another taxicab, another CO2, another taxicab, another ATP, another taxicab, another taxicab. All right, now that looks really mean, but this looks a lot better. You guys ready? First step, we went through glycolysis, the big G up here. Glycolysis is when we broke down glucose and led to 2 pyruvates, 2 ATPs, and 2 taxicab NADHs. Then we performed the intermediate step. The intermediate step took those 2 pyruvates we made up here and turned them into 2 acetyl-CoAs. And we lost 2 carbon dioxides here, but we made 2 taxicabs. All right, if you just stopped here, you literally only made 2 ATPs. So you're like, that's not enough. So here you go, guys. I'm going to go into the Krebs cycle, the big K over here. And in the Krebs cycle, I'm going to take my 2 acetyl-CoAs from up here, and I'm going to produce 4 CO2s. So I don't have any carbon left, guys. I used up all my carbons. Because if I take the 2 I lost here plus the 4 I lose here, that's my original 6. I got no more carbon at this point. But I've made 2 up here and 2 over here, so 4 total ATPs. I've also made 6 NADHs plus the 2 up here, so I'm up to 10 NADHs. And I made 2 of these other version of taxicabs. You guys have seen, you know, you go to the airport and there's like the yellow cabs, and then there's like these really cool green ones now. Like the NADHs are the yellow cabs, the more common ones, but the FADHs are like the green taxicabs. They're both taxicabs, they're just different colors. Okay? All right, so what I have yielded to this point, if I just stopped at the Krebs cycle, everybody, I have made 6 CO2s, I have made 4 ATPs. That's not a lot of energy. Like 4 ATPs, that's it? I went through all those steps for 4 ATPs. Are you kidding me? This is like, I don't know what's wrong with this. Why is this even done? Because 4 ATPs is hardly anything. I need millions of ATPs for each minute my cell is alive. So I got to bump this up a little bit. But don't forget, guys, I've got some taxicabs. Okay, so what do I need to do next, everybody? If you follow the steps, there was glycolysis to intermediate, intermediate to Krebs, Krebs to the electron transport chain, sometimes called the respiratory chain, guys, sometimes called the electron transport system. If you hit the Google on this one, you can find it under a variety of names. All right, guys, what did I end up with that was useful over here? 4 ATPs, cool. But I had a bunch of taxicabs, and here's where I'm going to ask those taxicabs, because these guys are not going to do substrate level phosphorylation. They're going to do oxidative phosphorylation for me. And here's where I'm going to put these taxicabs to work. So they've been shuttling electrons for me all along. You want to see what they do with those electrons? It's magic, everybody. All right. There's that NADH. It's carrying an electron for me. It's actually going to release that electron, and along the way, you're going to form a proton for that hydrogen. So you have a proton, and it's going to pump that proton across the membrane. And as you pump the protons from one side of the membrane to the other, they're just going to simply move through a form of diffusion across the membrane. So they're going to be more concentrated on this side, less concentrated on that side. But as you move down, all of a sudden, what did you end up having? You had you have a population more on this side now than on the other side. So literally, there's more hydrogen at the end of all that hard work on the outsides of your cell than there are on the insides of your cell. And if you're a bacteria, the only membrane that you can do this on is your plasma membrane. So this is the only membrane that a bacteria has to do this with. So bacteria use their own plasma membrane to make this happen. And they move protons across the membrane and create a gradient. And what do we know about gradients, guys? What do you want to do? You want to go down a concentration gradient. So they create a gradient, then they capitalize off of it. They create a gradient, they make a gradient, and then they capitalize off of it. How do they capitalize off of it, guys? Well, here I want you to think about, I'm going to go with, I don't know, let's see. I could do hydroelectric power. I can do wind power. Oh, let's do wind power. You guys good with wind power? Have you guys been to California? You're driving off the 10 and you guys are near Indio and all that stuff and you see all those really cool windmills. Okay, so you've got a version of a windmill embedded in your membranes. It turns out that is a specific pump called the ATP synthase. And since it ends in ACE, what does that mean it is? If it ends in the letters ASE, that must make it N. If it ends in the letters ASE, it's got to be an enzyme. Okay, guys. So remember, what did you do? You created a gradient where you have more hydrogen on the outside, less hydrogen on the inside. And so that hydrogen is going to flow down its concentration gradient. But guys, we're going to consider that flow of electrons kind of like a wind current. Flow of protons, excuse me. Flow of protons kind of like a wind current. And you know how windmills work? Windmills work because the wind creates a current that causes the turbine to spin. That turbine spinning is connected at the base to an energy harvesting, basically a battery. And that battery gets charged up every time this spins. All right, guys, well, guess what? You got a windmill in your membrane. You pump the hydrogen through at such a speed that you create a current. That current, literally, guys, you know what's the neatest thing ever? Is this molecule at the base of it, it looks like a windmill. I swear to gosh, it looks like a wind turbine. And this spins. It actually spins on the surface of a membrane. I am like, you guys don't believe me? I promise you, they have proven that it spins. And so as the hydrogens pump through, this thing spins and it produces ATP, pumping out the energy, keeping the lights on, guys. This cash money is a consequence of this turbine. Coolest thing ever. You never knew you had windmills attached to your membranes, did you? Well, ATP is produced and those hydrogens get into your cells. Now those hydrogens are in your cell. And what are hydrogens in a cell? Oh crap, guys. You know what hydrogen is once it gets into your cell? It's an acid. Uh-oh. Did you just pump an acid back into your cells? You don't want that acid in your cells. Well, you know what you did? Along the way, what else did you do, guys? You were breathing the whole time, weren't you? And your cells had some oxygen waiting on the inside for that hydrogen. And when that oxygen was hanging out on the inside waiting for that hydrogen, you made water. And that's how you avoided the insides of your cells from becoming acidotic. So oxygen was required to receive these hydrogens and make water. This is why you need to breathe oxygen right here, guys. Did you notice you didn't need oxygen anywhere else in the pathway? You're like, where's the oxygen? I thought I needed oxygen. You didn't need oxygen anywhere but here. Now, there are some really cool microbes that can use other carriers like nitrates or sulfates, but these guys perform anaerobic metabolism because they don't use oxygen. Seriously, Waverly, this is the coolest thing ever. All right, guys, now bacteria, they only have one membrane. So you know where bacteria are doing this? They're doing this on their plasma membrane. They're doing a ton of other stuff on their plasma membrane too, right? They're doing membrane transport. They got a bunch of crap happening on their membrane. So their surface area is limited. How do we take advantage of that in eukaryotes? In fact, how do eukaryotes do this better? Well, if bacteria only have one membrane, you know what you have? You know what you have? You've got mitochondria, everybody. Mighty mitochondria. Here it is in action. Because you know what the mitochondria have? They have membrane, then they have folds of membrane, and then they have folds and folds of membrane, and they have an outer surface and an inner surface of their membrane, and they fold that membrane in what we call cristae. And guess what? Each of those folds, you can do the same thing and put a bunch of windmills. And so this is why your mitochondria is so amazing, because you don't need to use your plasma membrane. You've got a built-in set of membranes right here. Mighty mitochondria kicks the butt of bacteria. I'll tell you right now, this is how you survive, because you have windmills embedded in your membranes. All right, guys, so everything that we just learned ends up producing a lot of ATPs. Okay, so we're going to take a pause here, but I have my summary slide coming up in a sec, okay? But we're going to just take a pause here, and we're going to bring together some of the math so you can understand the total ATPs, okay? Right off the bat, guys, we remember if we just went through glucose, it's kind of lame. Glucose alone is lame. 2 ATPs, I can't survive off of that. If I went through just the Krebs cycle, only 2 more ATPs. So you're like, lame. I can't survive off of that. So I'm only at 4 ATPs. But those taxi cabs shuttled a ton of electrons, and I can generate sometimes, guys, and I'm going to promise you that these numbers change. Okay, here's how these numbers change. I'll show you the math on it in a second, but these numbers change because eukaryotes are so much more efficient. We can get a little bit more energy out of each glucose. All right, guys, so the total possible production can be up to 40. On average, though, it's usually somewhere around 36 to 32. You get 4 ATPs from glycolysis, 2 from the Krebs cycle. Oh, yeah, so 2 from glycolysis in total, but then once you add the 2 from the Krebs cycle, plus the 38 from electron transport, minus the 2 you use, gives you 38 right here. This is some weird, funky math, so let's really give you the way to break down the math itself. Okay, guys, if you imagine it, here's the simple production. We remember we went into electron transport chain with 10 NADH taxicabs and 2 FADH2 taxicabs, right? So this is the stuff we need to use for our next step to calculate the math. All right, so what we consider possible with those 10 NADHs is that we can typically get, I'm trying to get to the math. I have a math slide here, guys. NADH, on average, we can get about 3 ATPs. So if I had 10 and I can get 3 for each one, that means I'm going to get about 30 from just NADH alone. Okay, so NADHs, the amount I brought in, I can get around 30. FADH2 isn't as good of a taxicab. It's a little bit slower. It drives a little bit slower. So I had 2 FADHs, guys. I can get about 2 ATPs. So this is going to give me another 4. So along the way, if I calculate all of my math correctly, guys, I get 30 ATPs from NADH. I get 4 ATPs from FADH2. And I started off everything with the big G up at the top, glycolysis. You ready to do this one more time? One final time, and then we've got our net reaction to go through. Okay, ready? I started off glucose, everybody. I started off glycolysis with glucose. And in fact, the word glycolysis is like glucose lysis, breaking down glucose. And I yielded at the end of the big G 2 pyruvates that are C3s, 2 ATPs, and 2 NADHs. You with me? Okay, did that one once. Then I went to the intermediate step where I took those 2 pyruvates and I turned them into 2 acetyl-CoAs, and I popped off a couple of CO2s and 2 NADHs right here. All right, when I went from the intermediate step, my next step was to go to the Krebs cycle.
Where does the intermediate cycle happen? So like as I was looking it up and researching and going on CK12, I never found this intermediate step. So I only have like 8 NADHs were produced. So the intermediate happens in the cytoplasm, or does it happen in mitochondria?
Yeah, it happens in the cytoplasm. So I'll actually have that later, but okay. I'm going to tell you right now, glycolysis, because this is a ubiquitous pathway, happens in the cytoplasm. The intermediate step occurs in the cytoplasm. And you know what's neat about the intermediate step is it doesn't require the same enzymes. So that's why it's considered an intermediate step, because it doesn't fall always. Remember, pyruvate can be used for other stuff. It doesn't always have to go down this next pathway. That's why it's not included in glycolysis. However, if you are a bacteria, all you've got is cytoplasm. So Krebs cycle is going to stay in the cytoplasm for a bacteria as well. Then they're going to do all of this electron transport chain. There's the big E at the end, guys. And they're going to do all of that on their cytoplasmic membrane, so their plasma membrane. If you are you and me, though, guys, you're going to shuttle these events into your mitochondria, because your mitochondria are a location for it. But if you are a bacteria, cytoplasm, 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 plasma membrane. Because you're a bacteria, and for today we're going to focus on bacteria. But when we get over there to the human system, we'll go back and do the mitochondria. But I just wanted you guys to understand why the mitochondria is so awesome. It's just so darn cool because you got a lot of surface area and it pops out a ton of ATPs. All right, guys, so I leave this Krebs cycle with this net yield. I've lost all of my carbon backbone. I've only made 4 ATPs. Totally lame. I need some more ATPs. And now I have 10 NADHs. I got 10 of these taxicabs and I got 2 of the FADH taxicabs. So my taxicabs are going to go shuttle. And what we know is for every NADH, I can make approximately 3 ATPs. For every FADH, I can make approximately 2 ATPs. And so then I start pumping them out of the membrane, and then I spin my windmill, and next thing you know, I generate, from these, I generate 30 ATPs, plus the 4 ATPs from the FADH2, and I made 6 water molecules. Now, if I'm breathing, everybody, I needed 6 oxygens. If I'm not breathing, if I'm an anaerobic microbe, I'm not going to be able to use oxygen because oxygen is toxic. So the net yield to date at this moment, guys, really the end result is 6 CO2s. You got them up there. You got 6 waters from right here, and you got about 38-ish ATPs. That's the net yield to date. After all this hard work, that's what you made. I'm not complaining, guys, because guess what? I made 38 ATPs. That's what I really wanted. I mean, here's the thing, guys. It's like CO2s. You really want some CO2? Here's some CO2. I'll breathe on you. You got water? I got water all throughout my cell. Yeah. But you know what I really want? I really want some cash money, don't I? Cash money when it comes to the world of ATPs is what matters. So, yeah, I'm with you. It's a pretty only have a plasma membrane, which means they have less surface area for making ATP, so they are less efficient. Eukaryotes, you got those mighty mitochondria, which make surface area for days, so they're highly efficient. So prokaryotes generate less energy than eukaryotes, and that's why, going back to the enzymes, conservation is key. Right? You guys remember how important conserving energy was? They can't make enzymes unless they need them. All right, guys. Now, aerobic metabolism is rocking, but microbes find a way to survive even a world without it. We know we have anaerobes. Some anaerobes use different forms of carriers, so they can make a decent amount of ATP. But the anaerobes that I'm interested in you guys knowing about are my fermentators. Come check out my fermentators. And I love fermentators, everybody. Yes, Dana, great question. They say don't feed the bacteria, don't feed the monsters within. 110%, don't eat sugar when you're sick. Wait a second, though. Don't you feel crappy and not want to eat anything else, though? That's the bad side. But yes, feeding them more sugars is not necessarily the best thing to do, for real. So what are you recommended to do? Broth-based diets, protein-based diets, not too many lipids if you have a digestive side of it, and just a small healthy subset of carbs, right? We want to feed them carbs that are a little bit more complex because it's harder for them to break down. So you're looking for your fiber, everybody. Complex carbohydrates, the microbes take a while to break them down too, so fiber is best. All right, let's go ahead though and imagine a world without oxygen. In a world without oxygen, microbes can go down a different direction. They'll stop at pyruvate. And by the way, guys, by the time they get to pyruvate, they've only made 2 ATPs. You paying attention? So fermentation, they only made the ATPs in glycolysis. Once they hit that pyruvate, they go down the other pathway. And the other pathway can be two different things. One, lactic acid fermentation. That's what your muscles can do. Or two, alcohol or acid fermentation. So you really only have two pathways to go through at the next step. All right, so fermentation, you start with glucose, you make your two pyruvates, and on one side of things, fermentation can take that pyruvate molecule, turn it into acetaldehyde, and then take that acetaldehyde and turn it into ethyl alcohol. Does anyone know what ethyl alcohol is? No, that's isopropyl alcohol, but actually we use fermentators to help us produce that. You know what ethyl alcohol is? That's ethanol, baby. That's booze. This is how you make your booze. You know what though? Isopropyl alcohol is made by bacteria too. Dom, I just want you to know, we can also make an isopropyl. So you know that rubbing alcohol? It can be made by bacteria if it needs to. All right, guys, but here is the booze pathway. So this is like the booze cruise, alcohol fermentation on this side. But in your cells, instead of going down that, guys, you take your pyruvate and you actually switch around the molecules a little bit. There's some enzymes in place. So if you don't have oxygen, you produce lactic acid. Lactic acid fermentation happens in your muscles. Everybody knows that because when you go for a really hard workout, you build some lactic acid in your muscles. That's because your muscles were doing anaerobic metabolism. They were starved for ATP, so they made some, but as a byproduct of not giving them oxygen, they made acid. Dun, dun, dun. So lactic acid metabolism can happen in you and me. Unfortunately, guys, you can't do this one over here. That's probably a good thing because you'd be drunk all the time. Seriously, you'd walk around, you'd be like, hey, I'm gonna go exercise so I can get drunk. Woo, that would be kind of crazy. All right, guys, so bacteria can make alcohol, and yeast are really good at it as well. For example, yeast in most of these are what we actually use for our alcohol production, but several bacteria can do it as well, and those are going to be our anaerobic microbes. The pathways are varied, very, very varied, guys. There's lots of different approaches. They don't always have to end in one type of acid. Lactic acid is one pathway. Yeah, that's probably true. Acetic acid. Hey guys, we're talking about our vinegar family over here as also another pathway. In fact, you know, your vinegar is really made by bacteria. Actinobacters make it. Here are the different pathways. Certain ones can go down. And so yeasts are definitely the ones used for alcohol production. Also, we use a lot of yeast and we use a combination of bacteria when we make cheeses. I was talking, you guys watched that video that the lady talked about eating more fermented foods, right? So you're going to have also the same ones that are going to do that to help you make kimchi or I'm sorry, there's even rotten eggs in Asian cuisine. There's also pickles and sauerkraut, right? So all of those fermentators take on and produce different types of acids along the way. But it's yeast that you're going to use to brew your beer and make your wine, okay? Or your vodka, or your brandy, or your whiskey, or your scotch, or whatever the heck you like to drink. Thank some microbes. For the next sip that you choose to take, whenever it may be, hopefully not right now, thank some microbes when that happens. Now remember, inside you and me, lactic acid is possible. And then there's all these other cool ones, but I'm not going to make you sick. But here's Clostridium making isopropyl alcohol, which is rubbing alcohol right there. All right. Now, foods when it comes to these guys are important. So when you're making cheeses and things like that, that fermentation, ethyl alcohol on one side. I can tell you that we use mixed alcohol to make acetic acid, propionic acid, that's food storage, by the way, guys. And then I got to gross you out about the fact that Clostridium is most famous for becoming anaerobic under the surface of your skin, and it's Clostridium perfringens that leads to gas gangrene, right? So you guys know fermentators can even take up residence under your flesh. In areas of your body where you have hypoxia, and maybe even subsequent anoxia, these fermentators can start to grow, and that's exactly how gas gangrene actually takes over. All right, so kind of cool. Okay, guys, so remember, what did we just eat? What did we start it off with and say that our metabolism began with, that the majority of the metabolism we have starts with? We said it all begins with glucose, right? Our Nike swoosh started us off in the world of glucose. Well, here's the whole thing though, guys. What if glucose isn't available as a food source? What if you've been doing a keto diet and you don't have any glucose? What if your bacteria are hungry and there's no sugar around? How do they survive? Well, just because glucose is the primary starting molecule, we can introduce other molecules and get you to the midway step. So for humans and for bacteria, the next preferred source of energy is going to be fat. In fact, guys, this is why you store love handles. Love handles are your storage source for energy, because fat is actually a fantastic source of energy. It's actually better than glucose is. But ironically, guys, you don't use it first. If you would just use your fat first, we'd all be skinny and amazing and happy. Oh my God, wouldn't that be lovely? Oh God, it would be. You do have a lot of energy. It's all stored. The problem is you can't eat anything to access it. You got to starve your body of the other nutrients that you're giving it, because they'll use the ones from your tummy before they'll use the ones from your love handles. Sad but true. So stop eating carbs and you can force yourself into lipid metabolism, which is the function of Atkins, South Beach, keto, all low carb diets. They're trying to force your metabolism to using lipids. Well, lipid metabolism breaks it down, and there's a specialized name called beta oxidation. Those of you in my A&P class, I'm going to show you all about that later. But lipid metabolism can use pyruvic acid, and in fact, you produce enough pyruvates that you go through the Krebs cycle more times. So for every carbohydrate every single gram of carbohydrates you consume, you get about 4 calories of energy. For every single gram of fat you eat, you get about 9 calories of energy. Like, that's more by a lot. That's more than twice the amount. So fats are really delicious calories. They're really high energy calories. Unfortunately, we don't use them first. The other thing, guys, it turns out we can actually break down proteins if we need to. And proteins can be broken down and they can enter into the cycle. Here's alpha ketoglutarate, which enters into the Krebs cycle and breaks things down as well. But you know why you don't want to use proteins for energy? Because if you get to the point where you're using proteins for energy, you are starving. If a bacteria, not just muscle loss, it's true starvation. So that's actually it. It's muscle loss for sure, but eventually, guys, you're using off that, you are in starvation. And we're going to learn about this. I'm going to make you guys sad because I'll show you pictures of starving people just to kind of bring in the downside. Okay, guys. So we learned a lot. Here's our summary slide from anaerobic and fermentation. Pyruvate itself generated from glycolysis can go through anaerobic metabolism if oxygen is not present. So remember, anaerobic metabolism and fermentation happen when oxygen are not present. That's how anaerobes survive. And our fermentators only generate the 2 ATPs from glycolysis. But when they actually make those, go through making those acids, they generate a bunch of CO2. In fact, guys, if you think about this, one of the reasons why bread will rise when you activate the yeast and put the yeast in, then the bread rises, is because there's pockets of CO2 made by those fermentators. You know where you get Swiss cheese? Swiss cheese happens when fermentators put pockets of CO2 into the cheese. Seriously, next time you eat Swiss cheese, you're gonna be like, wow, those holes were once filled with CO2. I don't care, I'm still gonna eat Swiss cheese. I'm like, yum, delicious fermentators. All right, guys, so what do we get? We get 2 types of fermentation. Lactic acid fermentation in us gives us sore muscles, and that's because we started doing anaerobic metabolism. Alcohol fermentation is how we make all the good stuff: wine, beer, cheese, vinegar, or unfortunately, medically, this is how those Clostridium grow. Woo! How you guys feel? You're like, I'm exhausted, Dr. C. Guess what? We are going to take a break. We're going to take just about a 10-minute break. We're going to come back together, and I'm just going to, I don't even think I'm going to do this one. I think I'm not going to teach you this one. Not today. Not today. Nope, nope, nope. I know. I'm debating it. I'm debating it because I'm like, you guys don't need it yet. I just want to show you guys that you build sugars up. You make glucose and gluconeogenesis, and you break it down. You make it glycogen and glycogenesis. But I'm teaching this to most of you, and if you've had metabolism in A&P, you learned it there as well. So I'm officially going to call this right here the stopping point, the breakdown side of metabolism, the stopping point for your next quiz, and for this chapter, the last that you need to know. Woo! Mic drop. Okay, so go take a break. I'll see you guys back here. Let's come back in 10 minutes or so. So let's go with seven after. And then when we come back, we'll talk about the quiz for next week and get some of you out of here and bring the rest of you in to do an A&P review sesh. See you guys at 10:07. right. Hi, everybody. Welcome back. So I want to be timely with this. I'm just going to take a brief moment just to remind you of what's coming up next week. Then I'm going to just go ahead because it feels better to introduce some of these concepts to you sooner. I'm just going to show you the last little bit of this chapter. Actually, I think I'll start there. So the last little bit on this chapter talks about, well, I should clarify, the very end of this chapter goes into photosynthesis. But as you guys remember, I'm not going to require any photosynthesis. But there's a small section at the end of this chapter that just discusses the upwards trend on the swoosh. So the anabolic pathways, sometimes called the biosynthesis pathways. And it's going to be important that you guys know these happen, but the stuff on the swish is actually all I test you. So that's why I'm okay with that. But I just want to show you that amphabolism, the process of moving things up, also occurs. In fact, in a scenario where a cell runs out of glucose, you can actually go the other direction and build the necessary molecules, starting with a precursor molecule like acetyl-CoA, pyruvate, or even that glyceraldehyde 3-phosphate. Things can be built from your cells. Starting with carbs, guys, it turns out you can actually make glucose if you need. The process of producing glucose is actually a really energy dependent process, but what we call that of making glucose from scratch, we call that gluconeogenesis. That process of making glucose from scratch is called gluco, sounds like glucose, new, to generate, genesis. The other side of it is if you make some glucose and you've got enough extra glucose you want to store, you can take that glucose and store it in the form of glycogen in something known as glycogenesis. So these are actually important processes, but I'm going to talk about them for my A&P students in the A&P class extensively. So it's good for you to see them now. You knew all along that this could happen though, because the Nike swoosh told us that we can go from precursors to building blocks to macromolecules up to a cell. So even though this last section is one that I don't test you directly on, you do need to know the concepts of the Nike swoosh. So let's speak about next week now. What you have coming up next week is quiz number 3. That will be on Tuesday, which for those of you in A&P, you're like, oh man, I got an exam on Monday and a quiz on Tuesday. I'm sorry, guys. It happens a couple times this term. I'm doing my best. So because of that, though, your CTQs are not due until Thursday. I've just published grades on those CTQs so you guys can see. Here are a few small things for feedback. A few of you got like a reference list, but didn't include citations. A few of you gave citations, but forgot a reference list. So you want to work on both. Other points of feedback, guys. APA style is something I'm trying to help you with. And APA style, I will not require it until the 4th set of CTQs. I won't start docking points until then. So I'm trying to use these first set to give you feedback, but I need to see a sign of improvement. So you guys, when it comes to APA style, the elements I'm looking for and the elements that should be include a title page. Right now, headers has been removed from APA 7th edition, but you do need page numbers and you do need double spaced font. Another thing I saw on some submissions, I saw the font, like it would be Times New Roman and then it would switch over to Arial and it would switch over to something else, guys. You want to make your font style consistent. And if you're switching fonts, it's a trigger to me as your instructor to think you're probably copying material. Okay, and that's not where we want to be. We want to avoid plagiarism at all costs, guys. So to avoid plagiarism, you need to cite your resources. If it is ever an exact quote from a resource, that's when you're going to not just cite the resource, but you're actually going to give us the direct page number where you pulled that quote. You don't need to put the page number unless it's a direct quote, by the way. Some of you put page numbers where you didn't need them, so that's another point of feedback. So APA style is something that students struggle with. It doesn't matter that you've had an English class, doesn't matter you did all your online classes. By the time you reach your nursing core classes, there's a bunch of you that may still not have writing in the correct format figured out. And I'm going to promise you right now, your instructors in future terms are going to expect that you know this. There is no excuse at this point that you don't. So I'm going to do my best with you guys is to try to reinforce it over the course of the term. So when it comes to APA style, guys, I am looking for an APA formatted title page. I'm looking for page numbers on your document. I'm looking for double spaced text. I am looking for citations in text. If it's a quotation, which means, guys, your similarity scores should not be flying red for me. And you know what? There was a bunch of your similarity scores that were dinging in the yellows, oranges, and reds, which meant that a good portion of that material was not your own words. And I can run them. If I actually suspect a student is plagiarizing, I have the analytics. It runs a score for me, and I can look at the similarity to your tools. I can evaluate where your tools are, and I can zero out your grade. And I will zero out a grade if it's blatant, but hopefully I can count on you guys not to plagiarize. So how do you do this important thing? It's called CYA, let you guys figure out the meaning of those letters. How do you CYA? Well, you know what you do? You put a citation and a reference. It's one way to cover yourself. And then you never directly copy from a single resource, guys. You know what? There are your peer group got through me. I taught them the same stuff I teach you guys. And as it turns out, all right, I'm going to look at that in a sec, Waverly. I just wanted to check if it was related to this. But as it turns out, guys, they still get over there. I've had students who are in future terms that have been dinged for plagiarism because they didn't know how to take materials. So I'm not saying to use me, guys. I'm not going to tell you like use me, but use me, because guess what? I'm not going to give you an F right now, but future instructors will give you an F if you make too many fallacies. So if I can help you build these skills, I've embedded some feedback if it was blatant. You guys just take a look and make sure you're working on better improving this. So you have one more submission that's due next Thursday. A week from today. It's over the chat. It's the week 7 ones. It goes up to chapter 9. And then you guys, and that's only a 15-pointer. And so try to make sure these elements are in there. If you've already turned it in and these elements aren't in there, add them and submit again. Okay, because we're trying to get you there. We're trying to get you to the point where you know how to do this so that you don't lose points in the future. I'm going to allow you guys 2 more mistakes, and then the last 2 of the semester, I'm sticking to my rubric. If APA is not there, if citations are not there, if plagiarism is in place, you will get a 0. If I catch plagiarism on one of the answers, the whole thing will get a 0, guys. Plagiarism is plagiarism is plagiarism. So I'm warning you now. I'm providing positive feedback to you guys so you can improve off of that feedback. But I'm telling you right now, guys, by those last 2 CTQs of the semester, you will get an academic violation if you plagiarize. I hope that scared you enough to make better habits, okay? And you know what an academic violation is? It's a 0 in this class. It's a documentation on your record. It's an actual documentation on your record. So any plagiarism, and I hope this scares a few of you because a bunch of you guys were close this last time. I won't even lie. And this next batch of answers does take a little bit of work. So remember to reach out for help. We can actually go through, after you guys finish your quiz on Tuesday, we can go through the questions and see if you have any before you submit. Okay, so I'll work with you. I'll help you in every way I can, but I don't want to see you taking the easy route, which is cheating. Okay. All right. So next week, quiz number 3, CTQs are due on Thursday. Reading chapter 9 sounds unrealistic to you guys, probably because you're going to have to read chapter 9 while studying for the quiz, and it's going to be a little overwhelming. Did you guys think metabolism was a challenging concept? You guys overall, even though we broke it down slow, it's probably one you're going to take some time to study. I think chapter 9 is harder or easier or just about the same. I don't know. It's my favorite stuff on earth. I can't even tell you what I love more than talking about genetics, but at the same time, it's tough. Okay, so if you can read ahead, cool. CK12, I won't lie, guys, I was pretty disappointed. I sent a bunch of you there, but it looks like some of you use some other tools. So you guys, maybe even if all you do is use a tool or 2 to try to introduce the concepts for chapter 9, but definitely it's going to hit you a little heavy. Now, you will not have a quiz on chapter 9. You will only have chapter 9 questions on the midterm. So that's another thing to pay attention to, guys. You don't have to quiz on chapter 9, but the midterm is only a week and a half away after that. And so what I've done for the midterm, guys, is I've pretended like your midterm is on a Tuesday, but in reality, I plan to push it to a Thursday, give you guys a whole week to study for your midterm, which is me being nice. None of your other instructors will ever give you extra time. They'll just push a midterm on you. So I am trying here, guys. But yeah, I want you to be aware that you won't get a second opportunity to quiz on this. So you got to be ready with those concepts by the midterm. But I'm going to send anybody who is not in A and P away unless you have questions. I'm going to send you away with the assurance that I'm going to pre-film a review session just like I did last time, and I'm going to publish it up there for you guys later. Contact me with questions, concerns. If you're in A and P, I'm going to open up the exam on a different screen and answer some questions for you guys. Sound like a deal? All right, so I'm going to leave you with this. Keep your eye out as an announcement. You too, Miss Jordan, keep your eye out for an announcement because I will pre-film a quiz review session over chapters 7 and 8. Sure, Nicole, I will answer that private message to you in a little bit. Waverly, do you want me to answer your question to in front of the group a little bit, just in terms of that, just to make sure you're on the right track?
It doesn't matter to me. I found a whole bunch of resources on Pinterest and on other things, and I felt really good going into the class. I did some of this CK12. And then as we were going through it, like my numbers on that chart weren't adding up. And I felt like it was a pretty good visual representation of what happens. And I'm definitely more visual than I am just reading off of a slide. But I'm just starting to see like how some lines aren't really, it just isn't all matching up for me. So it may help other people, but it's up to you.
Okay, is it possible, can I show off what you did? Can I grab your JPEG and put it up on the screen?
Yeah, that's fine.
Okay, so everybody, Waverly asked me a private question that I want to address before it goes too far. So hopefully I can help everybody by responding to this question. The answer that I have for you, Waverly, is that's actually very, very good math, and it's on point. I don't think ours is different. The problem is that, and ours really isn't different. What the problem is that the eukaryotes are more efficient than we are. So you can fiddle this math a little bit and get all the way up to 40, but 36 or 32 are the most standards. Your textbook uses the number 38, so I use the number 38, so it's consistent with your textbook. Okay, so what you did is probably what most people's math actually is, but your textbook uses that 38. Okay, guys, here's the whole thing. When I say you get 3 ATPs from NADH, that's an approximation, right? So none of this is exact, perfect math. So that's why there's a range. I'm going to tell you right now, most people put the range from about 36 to 40 ATP. So if your range is anywhere in that range, you're good to go. I need you guys to understand more specifically, you get 2 ATPs net from glycolysis, where the molecules are, and then you're going to understand that NADH and FADH2 are the major reasons why you transport electrons, and it's that ATP synthase whirl, the windmill that makes the ATPs. When it comes down to the total math, guys, be pretty happy with the math. But Waverly, I'm just like you. This is how I would draw the same thing. I am limited because I'm sitting here at home, and when I try to draw things with my mouse, it makes me mad. If I was in person, guys, I would have had each and every one of you up on the board, writing on your boards on your desk, and we'd all be drawing this together 8 times, okay? That's the way you learn it. But I like what she's done here. So let's follow what she's done so that you can see that she's right.
That's actually not the one that I made. That's the one that I found on Pinterest. Okay. The one that I made, I sent you second, and it doesn't show up as a JPEG. I think it just shows up as Chapter 8 Fillable Study Guide. But if you click on the image, but that's the one I started from. But I didn't understand how it said that the Krebs cycle is also involved in aerobic.
It is.
I mean, aerobic.
Aerobic. Remember, you can still go through the Krebs cycle if you have electron collectors on the anaerobic side. I was nice to you guys, though. I didn't make you do the anaerobic side fully. I just did fermentation. Do you remember me attempting to show you that picture of metabolism to scare you guys? You remember when I was attempting to show you how absolutely petrifyingly horrible metabolism is? All of these things are true, ironically, guys. So in my undergrad, which yes, I can actually remember that long ago. Seriously, I'm pretty old. In my undergrad, which was not a hot minute ago, it was more than a hot minute ago, guys. The hardest class I ever took was intermediate metabolism. It was the hardest class I ever had to learn. And the reason why is because none of these pathways is just a one shot. There's a lot of arrows that cross over. So it's like there's one pathway, but you can jump to that pathway. And then if you have this enzyme, you can jump to that pathway. And if there's not enough substrate, you can jump to that pathway. It is just yucky murky. So you probably were looking at some resources that are true, but not what I asked you guys to know, just because I tried to simplify it a little bit. Okay, so your resource was a little more, it's true, it's 100% true, but it's a little more complex. So here is what you put together, Waverly, and you're welcome to stay online and talk as much as you need, but she's got some really cool math for you guys here, right? She's got the basic outline of the reaction, which is what we built together. And at the end, you generate ATPs. It's the number of ATPs that are called into question here. So start with glucose. Glucose gets broken down through glycolysis, and the product of that is those pyruvates. Now the pyruvates shuttle into the Krebs cycle. So you need this arrow here, as well as like maybe this arrow, take that away and shuttle the arrow here, because those pyruvates become acetyl-CoAs. Acetyl-CoAs shuttle into the Krebs cycle. Then the Krebs cycle produces the NADHs, and along the way, you've got those NADHs, which are actually what goes into the aerobic respiration cycle. Oh, wait, there's your pyruvate. I see what you mean. Okay, so your glycolysis, your Krebs is too high. You need to move your Krebs. You got to move your Krebs out of the way. Okay, so then the pyruvate goes to acetyl-CoA. The acetyl-CoA then goes into the Krebs cycle. The Krebs cycle generates the NADHs, the FADH2s. And this FADH2, this NADH2, the two ATPs you walked out of the cycle with are what gets your math up. And so it just depends on what you multiply that 6 by. Some people multiply it by 2.8. Other people multiply it by 1.7. Like, I swear to God, guys, it just depends on the organism. It depends on the efficiency of the nutrient. There's just so many nerdy factors that I don't even want to get into because it'll just confuse you. But the reason why you get a different number depends on what you multiply that guy by and that guy by. Okay, so your cycle is on point, but you want to put pyruvate up here and Krebs down there, okay? And that's it. Yeah, otherwise it looks really good. It can be confusing, these anaerobic processes. Fermentation, you got it right. We talked about alcohol fermentation and acid fermentation. And ethyl alcohol fermentation is one type of fermentation, but there's a ton of other alcohol fermentations. You saw the huge list. I just want you guys to know there's alcohol fermentation and acid fermentation. That's it. It's not like I'm asking you guys to memorize any of the types of fermentation specifically. The goal of fermentation, only 2 ATPs, is also the main takeaway. It means anaerobic microbes cannot grow very quickly. They grow slow as can be. Aerobic microbes can get some energy, but you know where your cash cow is, guys? If you are a eukaryote and you have a mitochondria, that's how you really start popping out the energy. I'll review this again in that quiz review session I do. I'll go through it again so you guys can watch it again. If you're struggling at all, let's say Ninja, I like amoebas. You guys might want to grab the Amoeba Sisters videos on this stuff. I think Hank does a great job, but he's fast, guys. He talks at Hank speed. So if you want to watch Crash Course on this stuff, you can, but he talks fast, so you may like Amoeba Sisters a little bit better. They go a little slower. All right, guys. So if this was a hard chapter, I know you guys are sitting here thinking to yourselves, yeah, this was a pretty challenging chapter. And then, like I said, keep your eye out because chapter 9 next week it's got its own. It gives this one a run for the money. All right, I'm going to go ahead and invite the other 2 students who are in our other class, and I'm going to switch gears unless there are any residual micro questions. Okay, and if they can make it, they can make it. If not, I'm going to clear my screen. I'm going to grab my slides. I filmed the review sesh. I suspect nobody's had a chance to watch it yet. Did anyone have a chance to watch it yet? No, I didn't think so. I tried to get it in by dinner time yesterday, but I knew you guys had class today and you needed to focus on class today. Okay, guys, so it's about an hour and 10 minutes review session. And I published it in announcements, which is the new way I'm gonna send you guys all my updates, remember? Yeah, I can do both. Okay, let me stop that. That's easier for me than cutting the video later, actually.